Hello everyone, I'm by names of Nhali Dushimerene, and I'm going to present for you a video about glycolysis, and this is a process by which glucose is changed to pyruvate. Thanks. Hello everyone, I'm by names of Nhali Dushimerene, and I'm going to present for you a video about glycolysis, and this is a process by which glucose is changed to pyruvate. Thanks. Hello everyone, I'm by names of Nhali Dushimerene, and I'm going to present for you a video about glycolysis, and this is a process by which glucose is changed to pyruvate. Thanks. So we are going to start on process of glycolysis, which glyco means glucose, and lysis means breakdown. So in glycolysis, glucose, which is a 6-carbon sugar, is changed to pyruvate. Okay, we are going to talk about glycolysis, which so glyco means glycogen and lysis means breakdown. So this is the breakdown of glucose to pyruvate. So our first step is glucose, which is a 6-carbon sugar, which is going to be turned to 2 pyruvate molecules, which each one will be containing 3 carbon, and this will produce energy in form of ATP and NADH. So when we are talking about glycolysis, there are 2 phases. So the first phase, which is glucose turning to glyceraldehyde 3-phosphate, and this means the energy is going to be added. So in this first phase, energy is being added, and in this second phase, which is glyceraldehyde 3-phosphate turning to pyruvate, energy is gained. So let's jump to the cycle now. So the first step is glucose, which is a 6-carbon sugar. It is going to be oxidized to glucose 6-phosphate by an enzyme called hexokinase, and in this process ATP will be turned to NADH, and this glucose 6-phosphate is converted to fructose 6-phosphate by this enzyme called isomerase. And jumping to the next step, fructose 6-phosphate is turned to fructose 1,6-biphosphate, and in this reaction it is catalyzed by an enzyme called phosphofructokinase. This is a kinase enzyme, and ATP is lost to produce ADP, and fructose 1,6-biphosphate is oxidized by an enzyme called aldolase to form these 2 products. First product, which is called glyceraldehyde 3-phosphate, and another one which is called the hydroxyletone phosphate. And as you see, this is our first phase. As you see, we did not gain any energy, we were losing ATP. We lost 2 ATP. So here energy is consumed in this first phase. Let's jump out to the second phase. So this is our second phase, where energy is going to be gained. So here we have our 2 compound, which is glyceraldehyde 3-phosphate and dihydroxyletone phosphate, and this can form a reversible reaction by isomerase, which will push to this compound. So we are going to talk about glyceraldehyde 3-phosphate. It is oxidized to 1,3-bisphoglycerate, and in this reaction it is catalyzed by an enzyme called dehydrogenase, and here inorganic phosphate is lost, which is PO4, and here NAD is turned to NADH, and 1,3-bisphosphoglycerate is turned to 3-phosphoglycerate by an enzyme called phosphoglycerate kinase. And here, as you see, we are going to form ATP. ADP is going to form ATP, which will be our first ATP. Here we are going to go on 3-phosphoglycerate, which we turn to 2-phosphoglycerate by catalyzed by an enzyme called phosphoglycerate mutase. And our second last to form our pyruvate, 2-phosphoglycerate, it is catalyzed by an enzyme called anolase to produce phosphoenol pyruvate. And this phosphoenol pyruvate is going to be converted to pyruvate by an enzyme called pyruvate kinase. And here we shall form an ATP, where ADP will be formed to ATP. And this is our second phase. As you see, we formed 2 ATP, we gained 2 ATP. So this is our formation of pyruvate, which is a 2-carbon compound. Thank you everyone for watching the video. Hello everyone, by names of Nhwald Shimerene. I'm going to present my second video about Krebs cycle, and this is the synthesis of Hello everyone, we are going to talk about the Krebs cycle. First of all, you must know that the Krebs cycle is used to generate energy through oxidation of this acetyl-CoA, and this Krebs cycle, it is taking place in the mitochondria of prokaryotes and the cytosol of prokaryotes. First of all, if you watched my last video of how we converted glucose to pyruvate, as you see here, a glucose which was a 6-carbon compound was converted to pyruvate through glycolysis, and here we formed 2 pyruvate compounds, which each one will be containing 3-carbon compound. So this pyruvate will be converted to acetyl-CoA by an enzyme called pyruvate dehydrogenase complex. And here we are going to lose 1 carbon molecule and a NADH molecule. So if we lose 1 carbon molecule, we shall reduce our 3 carbons to 2 carbons. So this acetyl-CoA is a 2-carbon compound. So we shall bring our oxaloacetate, which is a 4-carbon, combining with acetyl-CoA, which is a 2-carbon, which is catalyzed by an enzyme citrate synthase to form this compound citrate. This citrate will be having now 6 carbon, because if you add this 4 carbon plus 2 carbon, which will give a 6-carbon molecule. So this citrate will be oxidized to isocitrate by an enzyme called aconitase, and this isocitrate is going to be converted to alpha ketoglutarate, and in this reaction is being catalyzed by an enzyme called isocitrate dehydrogenase. And here we are going to form NADH, and 1 carbon compound will be lost. So if we lose 1 carbon compound, this was 6 carbon, so this one will be 5 carbon compound. And this alpha ketoglutarate is going to be changed to succinyl-CoA. So this is catalyzed by an enzyme called alpha glutarate dehydrogenase, and here we are going to lose 1 molecule of carbon dioxide, and we are going to form NADH. So this NAD is going to form NADH. So because we have lost 1 carbon dioxide, and remember it had 5 carbons, so this will be a 4-carbon compound, which is succinyl-CoA. So succinyl-CoA will also be oxidized to form succinate. So this succinate, this conversion is catalyzed by an enzyme called succinyl-CoA synthase, and GTP will be formed in this reaction. So succinate is going to be converted to fumarate, and this is catalyzed by an enzyme called succinate dehydrogenase, and in this reaction water is going to be formed, which will later form FADH2. Fumarate is going to form marate. This is another molecule. So this is a 4-carbon compound molecule. So this is converted by an enzyme called fumarase enzyme, and this marate is going to be converted to oxaloacetate by an enzyme called malate dehydrogenase, and in this reaction we are going to form NADH. So this cycle will still continue. Now we have formed our 4-carbon compound until we again meet the 2-carbon compound, which is acetyl-CoA, and do the circle. So we are going to go through the results of our Krebs cycle. So as you see, we have 3 NADH which are produced. This is the first one, the second one, and this is the third one. As you see, this is our 3 NADH, and you have 1 FADH2, which is this one, and you have 1 GTP, so this one you have seen them, and 2 carbon dioxide molecules. So in what we call electron transport system, that's where we are going to form ATP through this compound we have seen, NADH. Thank you everyone for watching my video.